Okay, good morning, everybody. My name is John Clusen. I'm the host for the seminar this morning. And the topic today is the general introduction to the Avadis NGS Analysis software system. And the goal of the seminar is to provide you with enough information about each of the three main NGS workflows so that it's easier for you to decide to investigate the software further. And we'll take you through the three workflows and also take you into the biological context. The speaker today is Tom DeBoer. He's a scientist and product manager here at Strand Scientific Intelligence. And for those of you that have joined us on the phone lines are muted at the moment, and we will unmute them during the live question and answer session. If you are joining us through the web broadcast, I would encourage you to find the chat function within WEBEX and send questions during the presentation so you don't forget about them to both the presenter and the host. And what we'll do is read those out at the end so that you can get those answers right away. I'll also copy those chat questions in case we can't answer them in the time allotted or that they're fairly technical. And so we will be able to follow up with you afterwards. So with that, Tom, I'm going to give you the virtual floor.
Thank you very much, John. So, as John said, my name is Tom DeBoer, and I'm the Director of Product Management for Strand Scientific Intelligence. And I would like to talk to you about our new product, Avadis NGS, which is our solution for the analysis of NGS data. In only a few years, the analysis of the sequence of the human genome has gone from a multi-million-dollar endeavor involving both the public and the private sector to something that has come into the reach of everyday scientists. Obtaining the sequence of the first human genome cost hundreds of millions of dollars when the first draft was published in 2000, and although the final draft was only published really 7 years ago, and many scientists are still of the opinion that it isn't even finished yet. Today, the cost of sequencing a complete human genome to an average of, say, 40x can be as low as $20,000. And many industry analysts have predicted that the cost will drop to a mere few $100s per genome in only a few years, making it feasible to start thinking about sequencing every human baby when they get born. These cost savings have been made possible by many technological innovations introduced by such companies as Applied Biosystems, Roche 454, Solexa Illumina, and more recently by companies such as Ion Torrent, Complete Genomics, and Pacific Biosciences. But these costs usually only count the cost in reagents and do not usually include the cost of personnel running the equipment and performing the library creation, which can be a substantial real cost as well. Although the cost of the storage and computing of the raw information produced by typical sequencing experiments has become a significant portion of the total cost of a sequencing experiment as well. The cost of sequencing has completely outpaced the cost of computing, represented by the famous Moore's Law, displayed here as well. But all these costs pale in comparison with the cost that is introduced when it comes down to analyzing the result of sequencing experiments. No life science researcher just sequences a complete human genome of 100 individuals for the fun of it, but they have real scientific questions that they want to get answers to. Questions such as, what's the molecular cause of prostate cancer, or can we predict which individual has the propensity to develop a melanoma? But in order to get answering these biological relevant questions, many researchers find themselves having to deal with esoteric command line applications, many of which require extensive knowledge of computing environments that have basically not developed since the 60s. And to get to the underlying biological relevant information, a researcher often finds themselves jumping from one program to the next website and back again, which often is accompanied by data translation steps and file format changes. But many biologists did not go into biology to become bioinformaticians, but they wanted to do real research. And this is why we at Strand Scientific Intelligence are proud to introduce Avadis NGS, NGS Analysis for the rest of us. Avadis NGS is an application for the analysis of NGS data for RNA-Seq, ChIP-SEQ, and DNA variant analysis, and is specifically designed for and by life science researchers. In the next few minutes, I would like to show you more about the product that we have released really only a few months ago by showing you some examples of how the product works and how it can help you answer real biological questions based on NGS data and without having to become an expert bioinformatician. Avadis NGS provides downstream or tertiary analysis for NGS data, taking in pre-aligned data. So primary analysis is the analysis that is performed by the NGS hardware platform and which controls the machinery that does the actual sequencing. The end result of primary analysis is usually some representation of the raw reads with some quality metrics associated with them. FASTQ data format is a common exchange format for this data, but other data formats are routinely used as well. Secondary analysis is the activity that takes the reads from the primary analysis and aligns these reads to the reference genome. Typically, this is performed by software systems that are provided by the vendors, such as Agilent for Illumina systems or Bioscope for Applied Biosystems. There are also a large number of alignment programs available in the public domain, such as PWA, SOAP, Bowtie, etc. But for most applications, we often hear ELAND is good enough or Bioscope is good enough. And in many cases, your core facility or sequence provider can perform the alignment step for you, so be sure to ask that. After the primary and secondary analysis steps have been performed, the data is ready to be loaded in Avadis NGS. And Avadis NGS takes in pre-aligned data, which usually is in the SAM-BAM standard format, and allows you to interrogate the data in a structured and interactive fashion. We at Strand designed Avadis NGS specifically for you, the biologist, who is interested in answering a real biological question and does not want to become a bioinformatics expert just to do the work. We have a dedicated team of scientists, and a large section of which have advanced degrees in the life sciences to ensure the tools we develop are relevant for your research. We've developed the Avadis NGS product on the same platform as we used for the development of GeneSpring GX. And many of you may know GeneSpring GX as the popular analysis package for array-based gene expression analysis from our partner Agilent Technologies. They sell and market the application, and we develop it for them. Since Avadis NGS is built on the same platform as GeneSpring GX, people who know how to use GeneSpring can immediately get started with Avadis NGS, since it has the same look and feel as GeneSpring, and many of the features behave in the same way. This avoids the painful learning curve that is often associated with starting to use a new tool and ensures that you get your results faster. For those of you that are current GeneSpring users, you will be happy to know that some of the NGS functionality that is currently offered by Avadis NGS will be integrated into the GeneSpring platform in the coming year. This will allow you to perform both array and NGS-based analysis in the same package and compare and contrast the results from different and orthogonal technologies, thereby furthering enhancing your research. As we discussed in some of the previous slides, Avadis NGS allows you to analyze next-gen sequencing data using pre-aligned data. We have chosen to support the SAM-BAM format as the main import format for pre-aligned data, since it has rapidly become the de facto standard as the file format of choice for aligned data. It's made popular by the 1000 Genome Project and is now being adopted by most, if not all, NGS vendors as the standard exchange format for their aligned data. For instance, in the Illumina version of Cassava that is due out in February, version 1.8, it will adopt the SAM-BAM format as the only exchange format, and relegating the export format to the status of internal exchange format only. So for historical data that is not provided in the SAM-BAM format, there are a number of converters available that will quickly and without loss of information convert these data files into SAM-BAM format. So Avadis NGS supports the three major NGS workflow: ChIP-SEQ, RNA-Seq, and DNA variant analysis or DNA-Seq. In the next few minutes, I will discuss each of these three major workflows in more detail. And for those of you who have not tried your hand on Avadis NGS, I invite you to visit our website at avadis-ngs.com, where you can register to receive a free evaluation copy that is fully functional for 20 days. And I will repeat this information at the end of the seminar for those who want to write down the address. So this slide contains a little more detail on the three major supported workflows: ChIP-SEQ, RNA-Seq, and DNA-Seq. Now, the major objective in ChIP-SEQ experiments is to find regions on the genome that bind a particular protein or complex, such as transcription factors, RNA polymerase subunits, or histones, etc. The ChIP-SEQ experiment workflow in Avadis NGS allows you to find those regions in the genome that have an overabundance of sequence reads aligned to it, while looking for peaks in the coverage using a variety of peak finding algorithms, such as PICS and MAX. For transcription factor, this can be followed up by looking for potential binding motif in and around those peaks by using motif detection algorithms such as GADEM. In addition, the genes that are close to the detected peaks are good candidates for being regulated by those transcription factors and can also be detected. The gene ontology and pathway analysis tools can then be applied to learn more about these sets of regulated genes and their respective targets. For the study of the transcriptome, Avadis NGS supports the RNA-Seq experiment type or workflow. In addition to the almost classical differential gene expression analysis using the normalized expression level data, Avadis NGS allows for the detection of alternatively spliced transcripts as well. And both known and novel splice variants can be discovered. And we also have functionality to discover completely new genes and transcripts. In many disorders, such as cancer, the genetic makeup of a cell is changed in such a profound way that two genes or more are sometimes fused together, resulting in a hybrid protein with potentially devastating effect on the cell, and these gene fusion products can be detected with Avadis NGS. The third and last supported workflow is the detection of SNPs and small insertion and deletions, or InDels, in DNA. SNPs and InDels are discovered and annotated with information from dbSNP or equivalent databases. In addition to the dbSNP annotation, the effect of a SNP or InDel on the coding regions can be assessed as well, resulting in such classifications as non-synonymous coding SNP and frame shift InDel, etc. Larger structural variations, such as large insertions and deletions, as well as translocations and other rearrangements, can be detected for those experiments that have used paired end libraries or made paired libraries. So the next set of 7 slides will provide you with some more detail on the ChIP-SEQ workflow of Avadis NGS. So this slide explains the ChIP-SEQ experimental procedure that precedes the data analysis in Avadis NGS. So you normally start with a cell that is in a particular state, either growth phase or disorder, for instance, and the cell is then treated with a chemical that results in the cross-linking of all DNA binding proteins, such as transcription factors and histones, etc. The DNA is then extracted from the nucleus and shared to a particular fragment size. The fragments that contain a particular protein is enriched by the utilization of an antibody that is specific for that particular protein. The crosslink is reversed with chemicals and the protein is washed away, leaving us with just the DNA fragments that the protein is bound to. The fragments are subsequently subjected to the NGS library creation steps and sequence procedures, and the reads that can then be mapped to the reference genome. The resulting aligned data can then be analyzed in Avadis NGS. After loading the data in Avadis NGS, it will present you with a workflow panel on the right-hand side of the application that is specific for the chosen experiment type, in this case, ChIP-SEQ. The workflow provides a logical order in which to perform the various activities that comprise the analysis of a ChIP-SEQ experiment, usually from top to bottom. And the workflow is divided in logical subsections as well, such as quality inspection, filters, analysis, etc. So the first order of business is to determine the quality of the data and the alignment that was performed. There are a number of quality metrics and plots that can be produced by clicking on each of the workflow links in the quality inspection subsection. Based on the quality inspection results, you can decide to remove certain reads or decide to remove complete samples from consideration using the workflow links in the filter subsection. After the filtering is complete, the analysis subsection contains a number of analyses that can be performed on this filtered data. in the case of doing peak detection followed by motif detection, for instance. And then the last step in the workflow is to determine the biological relevance of the genes by doing gene ontology analysis and or pathway analysis. The previous slide already showed you some of the visualization capabilities of Avadis NGS, but I would like to highlight the genome browser in more detail at this point. Here we are looking at a region of the genome that is enriched in reads from a fragment that bound the neuronal transcription factor NRSF. This data and the data shown in some of the other ChIP-SEQ slides come from a paper from Junction et al. Published in Science in 2007. So the genome browser is divided in a number of tracks, and in this case there are 3 tracks showing, but there can be many more tracks. The top track is the reads tracks, and this contains the coverage and read data, in this case for a single sample. The middle track shows a region track that contains regions that were detected by the PICS algorithm as containing peaks. And the third track below shows the annotation information for the genome, in this case the ensemble transcripts. Other annotations tracks can be added, such as the dbSNP track or the annotation information from the UCC or REV-seq transcripts. The read track shows the coverage at individual positions at this zoom level, and the coverage for the forward and reverse strand is colored differently, with the forward strand coverage being colored blue and the reverse strand coverage being colored red. The separation of the forward and reverse coverage peaks is very typical for a ChIP-SEQ experiment, and this separation forms the basis of the peak detection algorithm in Avadis NGS. Below the coverage plot, the gray bars represent the individual reads, and the little colored bars show the mismatch to the reference in that read. Due to the possibly large number of reads in an individual sample, each track can be scrolled individually, ensuring that all important information is always in view. So the result of the peak finding algorithm is a list of regions that represent the extent of the coverage peak, and is basically just chromosome name and start end coordinates, like a BET file, plus some score and p-value that reflect the confidence in the peak being real. The next step in a typical ChIP-SEQ experiment that focuses on transcription factors is to determine if any of the peak regions show an overabundance of a particular sequence that could be considered the binding site for a transcription factor. The GADEM algorithm used in Avadis NGS scans the sequence for each of the peak regions and reports any sequence that is present more often than would be expected by chance. The result is one or more short motifs, the result for one of which is presented here on the left side of the screen in web logo style. On the right side of the screen, the locations of the regions that contribute to this motif are listed, and the p-values to determine the confidence we can have in these motifs being found in this visualization are shown as well. One result of the analysis of the data from the Junction paper mentioned before is presented here. The non-canonical motif of the NRSF, or the NRSE protein as they call it, transcription factor, is shown below the motif that was detected by Avadis NGS, and while the motif that was found by Avadis NGS is slightly shorter, is identical. A motif of interest that was found in the previous step can be scanned across the complete genome, or a fraction thereof, to determine if there is any additional binding sites for the transcription factor in the genome. This results in a region list that will contain all putative binding sites for the transcription factor under study. So the next step in the analysis is to determine all the genes that are potentially regulated by the transcription factor. And this can be achieved by simply scanning the genome and finding all the genes that are close to these potential binding sites. Once a list of potential transcription regulation target genes is constructed, the annotation of those genes can be evaluated using the gene ontology analysis capability of Avadis NGS. The goal analysis will answer the question, are any of the genes in my list annotated with certain gene ontology terms more often as can be expected by chance? And the gene ontology analysis will show the row categories that satisfy this criteria, and an example is presented here. So the list of genes that were generated using the Johnson paper data analyzing the NRSF transcription factor was shown to be overrepresented by genes that were involved in a number of ion transport processes, which is not surprising, knowing that the NRSF transcription factor is mostly active in the brain and specifically in neurons. So now we have completed a ChIP-SEQ workflow. It's time to discuss another great feature of Avadis NGS, which is the hierarchical nature of the result storage. In Avadis NGS, the result of any filtering or analysis is stored permanently with the experiment and stored in an hierarchical and chronological order. This makes it very easy to see how you arrived at the results in your experiment when you return to your experiments in a few months from now, when you can't really exactly remember what you did and how you got those results. Here we see a typical ChIP-SEQ experiment and its results. At the top of the tree-like hierarchy, we see the object called all-aligned reads. This object represents the data that was loaded and contains all the reads and the alignment of those reads to the reference genome for all samples that were loaded, and that was 2 in this case, as we can see from the sample folder below. The next step we did is run the peak detection algorithm PICS, and the results were stored as a descendant or tree branch of the all-aligned reads object. We next ran the motif detection workflow step, which resulted in 2 motifs being detected, one with 132 matches and another with 15. And next we found all the neighboring genes that were found close to the peaks we detected. And we know this since this is the descendant of the peaks detected by PICS object. Interestingly, we did not run the scan motif workflow step, which we normally would do. As we move on, we follow this up with a network and pathway discovery step and ran a gene ontology analysis with a p-value of cutoff of 0.1. So this concludes the section on ChIP-SEQ, and we will now move to the next section on transcription analysis with RNA-Seq. For an RNA-Seq experiment, the starting point for Avadis NGS is a set of reads already aligned to the reference genome. Usually this is done at the core facility or service provider that provided you with the sequence results. If you don't have access to a core facility or service provider that can do this, we recommend some of the publicly available alignment services such as Galaxy Alignment Service. These services take a FASTQ data file and can run alignment programs such as Bowtie and TopHat, and the result of which can then be loaded back into Avadis NGS. In this case, we have loaded an RNA-Seq experiment, which is a subset of the Berger paper for which the reference is shown at the bottom. We have loaded 2 samples from the leukemia cell line and 2 samples from each from a melanoma cell line. For users of GeneSpring GX, this view should be a very familiar display, which is not surprising since Avadis NGS and GeneSpring are both developed on the same platform called Avadis. The profile plot here shows the expression value for each of the detected genes in a typical line plot. You can clearly see a number of genes that are not expressed in leukemia, and they are expressed in the melanoma samples. And the reverse seems to be true for a single gene that is not expressed in melanoma but is expressed in leukemia. So we have the usual set of differential expression algorithms that have been made popular by the array-based expression analysis tools such as GeneSpring. But in addition, we have some algorithms that are optimized for RNA-Seq data as well. But profile plots only really show half the story. We all know that it's not the gene that is expressed, but its constituent transcripts are. While showing the profile plot for each of the transcripts is feasible, it soon becomes difficult to visualize this amount of data. If we assume, as many have, that each gene can result in an average of 10 transcripts, we can expect to see over 250,000 transcripts if all genes were active at one point. Avadis NGS contains an algorithm that will determine which of the transcripts is differentially spliced, and the results of a typical analysis is shown here. We have developed a special gene-centric visualization to display the differential splicing results and the corroborating evidence for this claim, called the gene view. So the gene view shows all the transcripts, known and novel, for a particular gene, and in this case we've shown GLRX. The transcript model we used in this analysis shows 2 transcripts for this gene, differing only in the length of the first exon. One on the left, the exon on the left. The profile plot shows 3 lines, one in red showing the RPKM value for the gene, and one blue and one brown showing the respective RPKM values for each of the 2 transcripts. As you can see in this plot, the expression level for the blue transcript stays basically the same, while the brown transcript goes up significantly in the melanoma sample as compared to the leukemia sample. So the evidence we have for the differential splicing is captured on the right side of the slide in the partition RPKM and the partition coverage plot. So the partition coverage plot shows the coverage for the partitions with the coverage for the leukemia sample shown in red, while the coverage for the melanoma samples is shown in blue. The 2 transcripts only differ in the length of the first exon, when seen from the right, seen from the left. And as you can clearly see that the coverage line is much lower for the extended part of the exon in both conditions, providing us with the evidence that the longer version of the transcript does not show any change in expression from one sample to the next. Therefore, it must follow that the higher expression level we see for gene GLRX is caused by the higher expression of the other shorter transcript. RNA-Seq experiments also provide the information for SNP detection. Here we are looking at a zoomed-in region of an area where the reads are shown as gray bars with only the mismatches to the reference genome showing up as base calls. There are a large number of mismatches to the reference genome in this particular position for this particular sample, and when the number of mismatches goes over a particular amount, 20% by default, a special visual clue will be displayed on the coverage track, where the colors show the distribution of the bases, and in this case red for T and blue for C. Of course, this is merely a visual clue, and it's no substitute for a rigorous SNP detection algorithm. But when we perform the SNP detection workflow step, we do find that this SNP is significant. The SNPs are stored as a special region list that can be visualized in the genome browser, as shown here as well. The other sample, the leukemia sample, is shown at the top, and it does not show any evidence for a SNP at this position. So this may be a good SNP for a follow-up analysis. Avadis NGS also allows for the detection of novel splice junctions using splice reads in RNA-Seq experiments. In the reads on top, the reads are represented by gray boxes, and splice reads that span 2 or more exons are indicated by a solid line. When we look closely at some of the reads, we can clearly see that some of the splice reads show evidence for exon skipping, and show that the first and the third exon are sometimes spliced together. In addition, we see numerous reads that fall inside the info region as well. Of course, visual inspection is not a proper discovery tool for this type of analysis, and we have implemented an algorithm that detects these new splice junctions. So the novel splice junction is indicated here as a separate track in the genome browser as a result of the splice junction detection workflow step, showing a possible new splice junction between the first and third exon. In this experiment, we had chosen the more conservative REF-seq database as the source for the known transcripts. The REF-seq database only contains those transcripts that are rigorously checked and verified to be real transcripts. And this means that this new transcript has not been submitted to the REF-seq database. When we compare our findings with the UCSC database of transcripts, we find that this transcript is actually a known transcript in that database. Obviously, somebody or some other program has detected or predicted this splice variant before us, and this gives us confidence that whatever new splice variants we may find could be real. So this concludes the section on RNA-Seq, and I will now spend a few minutes on the third supported workflow, DNA variant detection, or DNA-Seq. One of the most common workflows for SNP detection is to use the Agilent SureSelect target enrichment approach and perform targeted resequencing. Targeted resequencing allows you to focus your sequencing power and, more importantly, money on a small and targeted region of the genome, very often the exon regions of the genes. This can reduce the cost of a sequencing run substantially and allow you to focus on those important areas and limit the number of false positives that will need to be followed up. Avadis NGS supports the targeted resequencing approach by allowing you to filter out those spurious reads that fall outside a targeted region by applying a filter. And in this example, we show an Agilent SureSelect sample in 2 different tracks, one before and one after filtering. So in between the 2 tracks for the same sample, we show the bait regions for this particular Agilent SureSelect kit, the Exon version 1 kit. So after applying the filter on region list workflow step, the reads that fall outside the bait regions can be removed and will no longer interfere with the downstream analysis, such as SNP detection. And the filter will result in a separate object, allowing you to perform the SNP detection both before and after the filtering, and later compare the results to investigate the effectiveness of the target enrichment. After the filtering step, the SNP detection workflow step can be run, and a typical example of the result is shown here. The table shows a lot of information for each SNP that was detected. So the first row shows a single nucleotide polymorphism, or SNP, for which there are 21 supporting reads for a total of 21 reads, which make this a homozygous SNP. Heterozygous SNPs can also be detected, of course. The next column shows that this SNP had a score of almost 89, and that's relatively low. A higher number for this score in this column provides a higher confidence that this SNP is real. The calculation of the score is a complex algorithm, but it takes such things into account as the number of reads supporting the SNP versus the reference, the quality score for the base calls at this position in each read, and the mapping quality of the read, and a number of other criteria. So the SNP is a known SNP since we have annotated the SNP with the dbSNP identifier as well as some other annotations from the dbSNP database. The next highlighted row shows an area where more than one consecutive position is different from the reference, and this is shown as a MNP, or a multiple nucleotide polymorphism. And this MNP seems to be a novel discovery because we didn't find a dbSNP identifier. The next highlighted area shows that small insertion and deletions can also be detected with Avadis NGS. A couple of bases can easily be detected as well. In this case, it was also a known deletion. In addition to the information for the SNP itself, the table also contains information on the possible effect that SNP or InDel may have on the coding regions if it happens to fall in one or near one. Here's an example of a SNP that would result in a non-synonymous coding change, changing a lysine in an arginine residue for a particular transcript. So for each SNP and InDel that is detected, it's important to be able to verify the accuracy of the SNP call. There are various ways this can be done, and the score is one of them. And one other particular way we would like to share with you is through a specialized view we call the variation support view. In this view, it is possible to see a summary of the reads that support the variation call in a manner that's still compact and insightful. So in this example, we are showing a typical table of results and have selected a particular SNP we are interested in that seems to have a fairly good support. 83 variant reads and 47 reference reads. That's what 83/47 refers to. In the panel below, we're showing the clustering of reads that support the selected SNP. The top row shows the reference sequence in gray, and the position of the SNP is indicated in a light yellow column. The orange area either shows a dot when the read or cluster is identical to the reference in that position, or a base call if the read contains a base that is not identical to the reference. So the first line of the lower panel shows that there are 63 reads that show the variant base in the position of the SNP, but the rest of the reads are exactly the same as the reference as indicated by the dots. So the line is indicated as cluster 0, cluster ID cluster 0, since we effectively have clustered 63 reads into one read, representing them as a single line. The second line shows that there are 28 reads that are absolutely identical to the reference in every position, limited to the area around the SNP, which by default is about 20 bases on either side of the SNP. And there are various reads that support the SNP or the reference, but have other differences with each other and the reference as well. The number at the right-hand side indicates that most of these clusters are what we call singletons, and most likely represent some of a sequencing error, and not any sort of short-range haplotypes. But haplotypes could be detected with this view as well. In the case that an experiment contains multiple samples, a special SNP report is created that represents each of the positions for which at least one of the samples showed a SNP or an InDel, and the call that was made for each of the samples. In this example, a SNP was detected in sample 1, a CT heterozygous SNP, but the second sample did not show any evidence for a SNP there, and it was called as reference, so it was found to be the same as the reference at that position. In the second example, a homozygous CT SNP was found in both samples, although the score for both of them is fairly low, but it's there, and it's there in both samples. The third example, a SNP was called in the second sample, but there was not enough information in the first sample to make any call at all. That means NC, no call, and it's most likely due to low coverage or no coverage at all. So we couldn't make a decision one way or another. So the SNP detection workflow steps, as shown in the previous slide, allow for the detection of relatively small insertions and deletions, typically on the order of a couple of tens of bases. But larger insertions and deletions can be detected as well with Avadis NGS when using paired end or mate paired data, as shall be demonstrated by the examples here. So the colored blocks represent different locations in the chromosome or genome of a single sample compared to the reference sequence for the species shown below. The sample on the left can be seen as represent a large insertion when compared to the reference genome, and the sample on the right represents a large deletion when compared to the reference genome. If a paired end or mate paired library is created from these samples, the mate paired fragments will have roughly the same size or real mate paired distance in most cases, like 500 base pairs or something like that. So when we sequence the library and align those reads to the reference genome of the species, the apparent mate pair distance after mapping for the large insertions and deletions are different from the typical or average mate pair distance for the bulk of the reads. And assuming here that most of the samples are normal. So that this discrepancy in the apparent mate pair distance can be utilized in the detection of these large insertions and deletions in Avadis NGS. For large insertions, it's also possible that one of the mates does not map to the reference genome at all, for instance in the case of a completely novel insertion sequence. Or it may map to a completely different part of the genome. And this information can also be used to detect these large structural variations. And in a similar and related fashion, inversions and translocations can be detected as well. So here's a typical result of a large structural variation detection workflow, and the mix of deletions and insertions indicate the complex structure of this particular sample in this location, and this could be the basis for a follow-up study. I won't dwell on this one too much. Okay, so this concludes the section on the specific workflows for the 3 major supported NGS applications. And we will now cover some workflow steps that can be applied once a list of interesting genes has been uncovered. So Avadis NGS also provides network and pathway analysis functionality with information extracted from the scientific literature, which can be used to learn more about the genes that NGS analysis has shown to be affected one way or another. So Strand has developed technology that can extract information from the vast repository of knowledge that is the scientific literature. There are millions of papers that contain tidbits of important information that may not have made it into the traditional databases such as those in the NCBI or InDel repositories. Through the analysis by the natural language processing algorithms we have developed, we can turn an English sentence such as TLR-2 expression on monocytes was enhanced by macrophage colony stimulating factor MCSF and interleukin 10, but was reduced by transforming growth factor beta 1 into this, a graphical representation of the same information. Here the word expression is interpreted as a sort of regulation indicated by the blue box between IL-10 and TLR2. Enhanced is represented with the plus sign, and it also knows about the different spelling of the genes and proteins that can correctly identify MCSF as the same protein as CSF1. The information that was extracted from the literature like this can now be used to construct completely novel networks of interacting genes, or better said, proteins, as shown in this example. It shows the proteins and their interaction partners, which can include small molecules as well as complete processes such as cell death and cell division, etc. Even the typical location for proteins can be indicated as is shown in this example. So in this example, the green below is a representation of the nucleus, and the proteins in the cytoplasm are shown in the blue section, which represents cytoplasm. And the membrane proteins are shown on top of the cartoon of the cell membrane. So the proteins in the extracellular matrix are shown on the top. In addition to these novel networks, canonical pathways can also be loaded and displayed in a similar fashion. So pathways in the common OWL exchange format can be loaded and used to find significant pathways. And significant pathways are those pathways for which there is a larger representation in your gene list of choice than can be expected by chance. And that's in a similar way we do this with the gene ontology categories. Okay, so before we open up the floor for your questions, there are two more items I would like to discuss at this point about Avadis NGS. So the Avadis development platform that we use to develop Avadis NGS is a platform that was developed by Strand to be applied in a wide variety of application areas. We have applied the Avadis platform to such wide-ranging areas as array-based gene expression, like in GeneSpring GX, mass spectrometry, like in Mass Profiler Pro, and also a quantitative structure-activity relationship, or QSAR, in the program called Architect. So to ensure that the Avadis platform could be used in this wide variety of application areas, we adopted a design principle that will allow the application to morph itself, to adjust its look and feel based on the experiment type that you are analyzing. It will, for instance, only show the tools and icons that are relevant for that particular experiment type, and will not provide any tools or features that are not relevant or appropriate. Even within the NGS application, the workflow steps at the right-hand side of the application change based on whether you are analyzing ChIP-SEQ, DNA-Seq, or RNA-Seq data. So as shown here, the analysis section only shows those workflow steps that are appropriate for the type of experiment type and does not overly burden you with extra choices that are irrelevant or at least not common. So for instance, it's not typical to perform a SNP analysis in a ChIP-SEQ experiment, so the SNP detection algorithms are not shown by default in the analysis workflow section, while SNP detection is a very common step to perform in RNA-Seq and DNA-Seq. So they will show up in those workflow steps that support these experiment types. And one of the problems that many people struggle with in analyzing NGS data is to find all the appropriate annotation and sequence files. And Strand makes those files available within the application, which can be downloaded in a single click in the annotation manager shown here. So, and we also update these annotations on a regular basis, and you will be also notified when any of these have changed, so you can decide to update your annotations as well. Okay, that concludes the section on Avadis NGS specific features. I would like to invite you to visit our website at Avadis-NGS.com, where you can register to get a free trial version of Avadis NGS. And in addition to downloading the actual application, you can also download demo data in that same location. In addition to demo data, you can download annotation information as well. If a computer is not connected to the internet and cannot download the annotation data itself, you can put it on a little stick and move it to that computer, and then you can download it from that single file, again with a single click operation. There are also some example scripts that are more advanced tools that can be used for people who want to do more with Avadis NGS and maybe do some programming themselves. So that's going to conclude the broadcast section, and now it's time for us to answer any questions you may have.
Okay, this is John Klusman again, your host. I have unmuted the phone lines for those of you who have joined us by the teleconference. If you're joining us through the internet broadcast, you'll have to type your questions into the chat window. So I do have a first question from the chat, and the question is, what organisms are currently supported by the product?
There are, I need to get the right exact number, I think about 7 by now. So there's the human, mouse, and rat, Drosophila, yeast, zebrafish, and 1 or 2 others. So those are the ones that we currently have. As we said, we just released the product a couple of months ago, and we are still figuring out exactly which organisms people are working on. And you can load the data yourself, so if you have access to some of the files, you can definitely load them yourself. But you can also talk to us and send us an email at avadisngs.sales@strand.com and tell us which organism you are interested in, and then we can look to see if we can work with you to help you get going on those organisms. It's relatively straightforward.
Okay, and the next question is, are there workflow differences if you're working with single-end versus paired-end reads?
Somewhat. So, for instance, the structural variant detection will not run. It's still there, but it will tell you that your data does not contain paired-end data, so it will not even attempt to run. So it will check what kind of data there is. So there are some small changes here and there.
Okay, that's all the questions that were asked by the chat window so far. If you do have a question, please volunteer it now. We would love to be able to answer it live. And for the callers who do have the live open line, be happy if you wanted to speak up, just say your first name so we know how to follow up with you afterwards if we can't answer your question live. And go ahead and ask away. Okay, I hear everybody's waiting for somebody else to jump in with a question live here. So we'll just give it another 10 seconds here if someone wants to ask a question. Otherwise, we will thank you for your time and attention this morning.
Okay. Well, thank you, everybody. And hopefully this was insightful.